Hello there, and welcome to the Sleepy Science Channel. Tonight, we're stepping into a world that exists inside every living thing, yet is rarely seen or truly considered. Cell biology is the story of life at its smallest scale, where countless microscopic structures work together in unimaginable ways. They build bodies, thoughts, movement, and memory. Inside you right now, trillions of cells are active. They sense their surroundings and communicate with neighbors. They repair damage and make decisions that shape every moment of your life. This hidden world is not simple or mechanical. It is dynamic, responsive, and deeply creative. Cells construct, recycle, and dismantle, and can even sacrifice themselves for the greater good. They cooperate, specialize, and adapt in ways that rival any city or ecosystem on Earth. From the first moments of life to the quiet moments while you sleep, cells are always at work, carrying out ancient processes refined over billions of years. As we explore this topic together, you may begin to notice life differently, not as something solid and fixed, but as a living flow of activity, balance, and renewal happening far below the surface. If you enjoy these gentle journeys, I invite you to like, subscribe, or share a thought below. It helps others find their way here too, one sleepy soul at a time. But for now, all you need to do is relax. Let your body soften and allow your eyes to grow heavy, and allow your mind to gently unwind as we explore this microscopic world together. Let's begin. Your body runs on trillions of tiny cells, and each one is alive. Even when you are perfectly still, your cells are busy in ways that feel almost impossible to picture. Some are gripping to your bones and sensing pressure. Some carry oxygen on a one-way trip through your bloodstream. Some are scanning for trouble, while others are repairing where you never noticed. A few seconds can mean a whole new batch of molecules built, folded, checked, and delivered to the right place. Your body is not one thing. It is a community that stays coherent because countless cells cooperate, take turns, and follow shared rules. They grow, rest, respond, and sometimes step aside so the whole system stays healthy. When you think about life, it can help to start here. You are a living crowd that somehow feels like oneself. A single cell can be a factory, a city, and a library. Inside one microscopic space, there are workstations, shipping routes, security checks, and storage rooms. Raw materials arrive, and useful products leave in carefully wrapped packages. Energy is managed, waste is sorted, and building blocks are assembled with startling accuracy. At the same time, the cell holds instructions for making more of itself, and it keeps those instructions protected and organized. That combination is rare in the human world. A city does not usually contain the complete blueprint for rebuilding the city. A factory does not usually carry the full history of its design. Yet a cell does both. It can respond to a change in temperature, a sudden lack of nutrients, or a chemical warning from a neighbor, then adjust its inner work without waiting for outside help. Life at this scale is not simple. It is compact, coordinated, and endlessly inventive. Your cells talk constantly, using chemical messages like tiny texts. Communication is how a body stays coordinated instead of becoming a swarm. Cells release molecules that drift across short distances or travel through blood to reach faraway targets. Some messages are fast and local, and some are slow and wide-reaching. A signal can tell a cell to divide, to specialize, to move, or to stop and wait. In the brain, communication becomes a rapid cascade where one cell's message changes the electrical mood of the next. In the immune system, cells swap information about threats then call reinforcements with urgent chemical flares. Even bacteria have versions of this idea. They can sense how many neighbors are nearby, then change behavior as a group. The result is that life becomes more than individual survival. It becomes coordination. The amazing part is that the language is chemistry, and the meaning depends on context, timing, and the receiver. Cell membranes choose what enters and what never gets in. The membrane is built from molecules that naturally form a flexible barrier, and that barrier separates a controlled interior from the outside world. Yet the membrane is not a wall. It is more like a guarded gate, filled with proteins that recognize specific passengers. Some let water slip through. Some open only when a signal arrives. Some spend energy to push ions against a gradient, building tiny electrical differences that cells can use later. This selectivity is what makes life possible. A cell can keep harmful substances out, hold on to valuable molecules, and maintain a chemistry that is different from its surroundings. When membranes fail, the consequences can be dramatic. Toxins can punch holes. Viruses can trick their way in. The membrane is a quiet hero, making order in a world that wants to mix everything together. Many cells recycle their own parts, then build them again. Recycling is not just an environmental idea. It is a survival strategy inside living cells. When parts wear out or become damaged, cells can wrap them up, break them down, and reuse the components. This helps prevent clutter, and it also helps during scarcity. If nutrients run low, a cell can temporarily reuse its own materials to keep essential systems running. This process can clear out faulty proteins that might otherwise form harmful clumps. It can also remove damaged structures that leak reactive chemicals. In many cases, recycling is linked to resilience. Cells that manage waste well can tolerate stress better, and they can recover faster. The striking thing is that this is not random destruction. It is selective housekeeping with a purpose. The cell is constantly deciding what to keep, what to repair, and what to dismantle for parts. Life stays stable because it is always being renewed from within. Some cells self-destruct on purpose to protect the whole body. This is one of the most dramatic forms of cooperation in biology. A cell can receive signals that tell it to shut down in an orderly way. It breaks its contents into neat packages, and those packages are cleared away by neighboring cells or immune cells. This prevents messy spills that would inflame the surrounding tissue. Self-destruction helps shape the body during development. It can remove temporary structures and sculpt precise forms. It also acts as a safety system. If a cell senses severe DNA damage, it may choose to exit rather than risk turning into a dangerous runaway. Some viruses try to block this choice because they want the cell to stay alive long enough to produce more virus. Cancer can also involve failures in this process, where damaged cells refuse to step aside. Seen this way, self-destruction is not a tragedy. It is a protective rule that keeps a multicellular body from being undermined by a single faulty member. Stem cells can become many cell types when the signals are right. What makes stem cells so fascinating is not just their potential, it is their restraint. Many sit quietly in special niches, surrounded by supportive cells and chemical cues that keep them stable. When a tissue is injured or worn down, signals shift. The stem cell can divide, then produce one cell that stays a stem cell, and another that begins to specialize. Over time, those descendants can become the working cells of the tissue. This is how some parts of the body maintain themselves through years of use. The idea also reshaped medicine. Scientists learned ways to coax certain cells back toward a more flexible state, then guide them forward again. That opened doors to growing replacement tissues and studying diseases in the lab using patient-derived cells. It is not simple, and it is not magic. It is careful control of identity. A cell's fate can be rewritten, but only when the instructions, environment, and timing align. Your immune cells patrol like guards, checking every cell's identity. Your body has a problem that sounds almost philosophical. How do you defend against invaders without attacking yourself? One answer is constant inspection. Many immune cells move through tissues and blood, feeling for signs of danger and checking molecular ID tags on cells. Healthy cells display patterns that say, in effect, I belong here. Infected cells can display unusual fragments that reveal something foreign is happening inside. When that happens, immune cells can respond with precision, calling help, killing infected targets, and remembering what they saw for the future. This is also why organ transplants are so challenging. A donated organ can look like an intruder because its ID tags do not match the recipient. Immune patrol is not only about fighting. It is about maintaining order. It clears damaged cells, watches for early cancer changes, and keeps the boundary between self and not self from collapsing. Cells can crawl, even without muscles, using shifting internal scaffolds. Watch a white blood cell under a microscope, and it behaves like a tiny animal. It extends a broad leading edge, anchors briefly, then drags the rest of itself forward. The engine is not muscle tissue. It is a fast cycle of proteins that assemble into fibers at the front, then disassemble and recycle at the back. Some cells move with flat sheets that spread like a fan. Others use rounder bulges that pop outward, then grip and pull. This crawling matters in real life. Immune cells squeeze through tight spaces to reach an infection. Repair cells migrate into a wound and help rebuild the surface. During early development, migrating cells help place tissues in the right locations. The world inside your body is full of purposeful motion, and much of it is powered by single cells that can crawl with surprising determination. Many cells grip their surroundings, then pull themselves forward. Movement is not only about pushing. It often begins with holding on. Many cells form temporary grips that latch onto the material around them. Those grips can be so small they are invisible without special tools, yet they act like climbing hands. Once attached, the cell tightens internal cables and pulls its body forward, then releases the rear grips to repeat the cycle. This ability shapes how tissues heal and how organs take form. It also explains how cancer cells can spread. A wandering cell is not just drifting. It is actively testing surfaces, choosing where to attach, and hauling itself along. Cells can even adjust their grip strength. On some surfaces, they spread wide and pull steadily. In tighter spaces, they use smaller anchors and faster bursts. In a sense, every moving cell is an engineer, building and dismantling attachments as it goes. It is a quiet kind of strength, measured in tiny pulls that add up to real journeys. Cilia sweep fluid along tissues, like coordinated underwater grass. Some cells wear forests of microscopic hairs that beat in rhythmic waves. Each hair is tiny, but together they can move fluid with clear direction and purpose. In your airways, this motion helps carry trapped dust and microbes upward, away from the lungs. It is part of a cleaning system that works while you breathe, talk, and sleep. In the brain, cilia help circulate fluid through spaces that bathe delicate tissue. In the reproductive system, similar beating can help move an egg along its path. What makes cilia so fascinating is coordination. Neighboring hairs beat with slight delays, creating a traveling wave that is more effective than random flapping. This is not conscious teamwork. It is biology following physical rules, producing elegant patterns that keep tissues clear and fluids moving. A single cell can shape the flow of an entire microscopic environment. Flagella spin like propellers, pushing single cells through liquid. Some cells do not crawl or wave. They swim. A flagellum is a long, thin tail that can drive a cell through water by rotating or whipping in controlled patterns. Many bacteria use rotating flagella like tiny motors, and they can change direction by changing the spin. That lets them explore, retreat, or surge forward toward better conditions. In animals, sperm cells use a different style of tail movement, a flowing whip that pushes them onward through thick fluid. What is striking is efficiency. At this scale, water feels more like syrup than a pool. Momentum fades almost instantly. So swimming requires constant, carefully shaped motion. Small changes in how a flagellum bends can mean the difference between progress and going nowhere. In lakes, soils, and bodies, this propulsion turns single cells into travelers with destinations, guided by chemistry and chance. It is locomotion built from protein, tuned to a world where viscosity rules. Cells divide by copying everything, then splitting with high precision. Before a cell can become two, it has to prepare like a careful traveler packing for a long journey. It duplicates its DNA, but it also increases many of the parts that keep it running. Membranes expand. Key organelles grow and duplicate. Supplies of building blocks are stockpiled, so the two new cells will not begin life hungry. Then the cell reshapes itself, so the contents can be shared fairly. At the end, a tightening ring pinches the cell in two, sealing each half like a perfectly tied knot. When this goes well, the result is almost miraculous. Two cells step away carrying the same core instructions and enough internal equipment to survive independently. This is how wounds close, how tissues renew, and how a single fertilized egg begins the long road toward a whole living body. Many cells stop dividing, yet stay active for decades. Not every cell spends its life making copies. Many cells step out of the cycle of division and dedicate themselves to specialized work. A heart muscle cell spends its days contracting in steady rhythm. A lens fiber in the eye helps focus light with remarkable clarity. Many brain cells focus on communication, support, and maintenance rather than replication. This choice has benefits. A highly specialized cell can invest in performance instead of constant rebuilding. It also has costs. When damage accumulates, replacement can be limited. That is why some injuries heal poorly and why certain tissues are vulnerable to aging. Still, these long-lived cells are not passive. They adjust their chemistry, respond to signals, and repair daily wear. They can change how strongly they work and how they connect to neighbors. A cell does not have to divide to be alive and dynamic. Some of the most important work in your body is done by cells that chose stability over duplication. Some tissues renew fast, while others rebuild slowly over years. Your body is a patchwork of different schedules. Some tissues face constant abrasion, constant chemical exposure, or constant strain, so they replace cells quickly. Others are protected or built for endurance, so they renew at a slower pace. Blood is refreshed continuously because its job is to travel and to be used up. The lining of the mouth and the surface of the eye deal with friction and drying, so they renew steadily. In contrast, tendons rebuild slowly because they are dense and sparsely supplied with blood. Some parts of the brain are careful about replacement because wiring has to remain stable for memory and skill. This difference in timing is part of what makes biology feel so intelligent. The body is not renewing everything at the same rate, and it is not wasting energy where it does not need to. Each tissue has its own budget, its own risks, and its own strategy for staying functional across a lifetime. Your gut lining renews quickly because it takes constant wear. Every meal is a challenge for the cells that line your intestines. They face acids, enzymes, rough particles, and a dense crowd of microbes. At the same time, they must absorb nutrients and keep dangerous bacteria from crossing into the body. That is a high-stakes balancing act, and it demands constant renewal. Deep in the folds of the intestinal lining are stem cell-rich pockets that keep producing fresh cells. Those new cells migrate upward, take on specialized roles, and then are shed into the gut after a short working life. This rapid turnover helps keep the barrier strong even when damage happens daily. It also helps the gut adjust to changing conditions. Diet, infection, and inflammation can all alter what the lining needs to do. A renewing surface can adapt faster than a fixed one. It is one of the reasons the digestive system is both resilient and sensitive. The gut is not just a tube. It is a living frontier that replaces itself again and again. Mitochondria turn food into usable energy second by second. When you eat, you are not just filling a tank. You are feeding a chain of reactions that will be converted into a usable form of energy inside your cells. Mitochondria take molecules from food and strip away electrons in a controlled sequence. That flow helps build a gradient across an inner membrane, and the gradient powers a tiny molecular machine that makes cellular fuel. The beauty is the scale. This happens in countless mitochondria across your body, in a continuous stream. When you run, demand rises, and mitochondria respond by speeding up output. When you are cold, some tissues can turn more of that process into heat. When oxygen is scarce, the whole system has to adapt. The result is that your ability to move, think, and maintain body temperature depends on microscopic power plants working without pause. It is chemistry, but it feels like magic. Cells read DNA like recipes, then build proteins on demand. This is one of the most astonishing ideas in biology. A long chemical code can be copied into short working messages, then used to assemble proteins one piece at a time. Those proteins become the doers of life. They carry oxygen, send signals, build structures, and speed up reactions that would otherwise crawl. What makes it even more exciting is timing. Cells do not use every recipe all the time. They switch genes on and off depending on needs, location, and circumstance. A muscle cell and a neuron can carry the same DNA, yet make very different sets of proteins. Small changes in the code can change a protein's shape, and shape can change what it does. This is why tiny mutations can be harmless, helpful, or dangerous. It is also why biology can evolve new abilities from small edits over time. The nucleus stores DNA, but it also controls daily cell activity. The nucleus is often described as storage, but it behaves more like mission control. It does not just guard genetic information. It decides what gets used, when it gets used, and how strongly it gets used. Inside, sections of DNA can be opened up or kept tightly packed, and that choice changes which instructions are available. On the nuclear surface, gates called pores regulate traffic. Messages leave, and carefully selected proteins enter to influence which genes are active next. This is how the same DNA can support a liver cell that detoxifies and a skin cell that forms a tough barrier. The nucleus also responds to stress. If conditions change, gene activity can shift in minutes. It is a responsive system, not a dusty vault. A cell survives because its nucleus keeps adapting the plan. Cells can sense stiffness and react differently on soft surfaces. A cell does not just live on a surface. It measures it. When a cell attaches, it pulls gently and reads the resistance, almost like testing the firmness of a mattress. On softer material, the cell may stay rounder, move differently, and switch on genes linked to a softer tissue identity. On stiffer material, it may spread out, strengthen its attachments, and favor a different program of growth. This sensitivity helps cells build the right structures in the right places. Bone-forming cells respond to rigid environments. Cells in fatty tissue respond to softer ones. In the lab, changing the stiffness of a gel can steer what certain developing cells become. In disease, stiffness can change. Scar tissue becomes tougher, and that altered feel can influence how cells behave nearby. The idea is mind-bending. Cells do not only read chemical signals. They read the physical texture of their world, and they rewrite their future based on what they feel. Some cells feel touch through proteins that open under pressure. Touch begins long before the brain interprets it. At the surface of certain cells are proteins that behave like tiny gates. When the membrane is stretched or pressed, these gates change shape and open. Ions rush through, and that sudden flow becomes a signal the cell can understand. In your skin, these pressure-sensitive channels help convert a tap, a vibration, or a sustained press into electrical activity that nerves can carry inward. Similar sensors exist in places you might not expect. Blood vessels need to sense stretch as pressure changes. The bladder needs to sense fullness. Even some cells in the inner ear rely on mechanical forces to help translate motion into sensation. What makes this so captivating is simplicity. A physical push becomes chemistry, then electricity, then perception. The first step is a protein that can be opened by force. It is a reminder that your sense of the world is built from molecular shapes responding to pressure, one tiny gate at a time. Cells can sense chemicals and chase them, called chemotaxis. Imagine smelling smoke and moving toward safety, but at the scale of a single cell. Chemotaxis is the ability to detect a chemical gradient and move in the direction of more or less of that chemical. Immune cells use this to home in on infection. Bacteria use it to find nutrients or avoid toxins. The trick is that the differences can be tiny. One side of the cell detects slightly more molecules than the other, and that small imbalance is enough to bias movement. The cell takes a step, samples again, then adjusts. Over time, these tiny decisions create a purposeful path. Chemotaxis also shapes development. Growing nerves can follow chemical trails toward their targets, following connections with surprising accuracy. It is not a map you can see. It is a scent you can measure. In a world of drifting molecules, cells can hunt, escape, and navigate using gradients as invisible signposts. Cells can sense light, even in tissues far from eyes. Light is not only for vision. Many cells carry light-sensitive molecules that can change cell behavior when exposed. In the skin, light can influence pigment production and timing signals that affect daily rhythms. In the eye itself, certain retinal cells act less like image detectors and more like brightness meters, feeding information to the brain systems that set sleep and decay. Wake patterns. Even some organisms without eyes can respond to light. Single-celled algae can swim toward illumination to improve energy capture. What is remarkable is that light can act as a control signal. It can trigger gene activity, shift chemical pathways, and adjust internal timing. This gives life a way to sync with day and night using the most reliable cue on Earth. You do not need to be looking at light for it to matter. Your cells can treat light as information, and their responses ripple outward into behavior, mood, and physiology. Cells can sense oxygen levels and switch genes accordingly. Oxygen is both a gift and a constraint. Too little starves energy pathways, and too much can increase damaging chemistry. Many cells monitor oxygen using molecular sensors that trigger changes in gene activity when levels drop. In low oxygen, cells can shift how they make energy and increase signals that encourage new blood vessel growth. This response is crucial during wound repair, where oxygen supply can be limited until circulation improves. It also matters at high altitude, where the body must adjust to thinner air. Tumors can create low oxygen pockets too, and cells in those regions often change behavior in ways that affect growth and treatment response. The big idea is flexibility. Oxygen availability is not constant across the body. Cells adapt locally, tuning their metabolism, transport proteins, and survival strategies to match what is available. This is not a slow evolutionary change. It is a fast, built-in switchboard that helps cells survive shifting conditions minute by minute. Cells can sense nutrients, then slow growth when food is scarce. Growth is expensive. Building new proteins, membranes, and DNA requires a steady supply of raw materials and energy. Cells therefore keep watch over nutrients, and they make a choice when supplies drop. They can reduce protein production, pause division plans, and redirect energy toward maintenance. This is one reason fasting and starvation trigger broad changes across tissues. Cells shift from building mode to survival mode. In microbes, nutrient sensing can be the difference between rapid growth and entering a dormant state that waits out hard times. In complex bodies, the same logic helps protect cells during stress. When resources return, growth can resume, but the pause prevents reckless spending when the budget is low. The striking part is that this decision is built into the machinery of life. Cells do not blindly grow until they fail. They negotiate with their environment, reading scarcity as a signal to slow down, conserve, and endure. Mitosis lines up chromosomes, like a careful sorting machine. Imagine trying to divide a library of precious books while making sure every volume ends up in the correct new building. That is the feeling of mitosis at the microscopic scale. Chromosomes condense into tidy shapes so they can be moved without tangling. A structure made of fibers reaches out from opposite sides of the cell and attaches to each chromosome. Then comes a moment of tension, where everything is checked and held in balance. Only when the attachments are correct does the cell commit. The chromosomes are pulled apart with astonishing coordination, so each side receives a complete set. This is not just movement. It is choreography under strict rules, guided by checkpoints that can halt the process if something looks wrong. When people talk about life being orderly, this is one of the clearest examples. The cell treats inheritance like something sacred, and it refuses to rush. Errors in division can create extra chromosomes, changing cell fate. Most of the time, chromosome separation is exact. When it is not, a cell can end up with too many or too few copies of certain chromosomes. That imbalance can change the entire trajectory of the cell. The cell might struggle to grow because important genes are present in the wrong doses. It might trigger stress responses and stop dividing. It might be eliminated by quality control systems. Sometimes the cell survives and keeps going, and that survival can matter. In a developing embryo, chromosome imbalance can cause major changes in how tissues form, and it can contribute to pregnancy loss. In adult tissues, a cell with the wrong chromosome number may gain an advantage or a weakness, depending on the genes involved. This is one reason cancer can be so unpredictable. A small error during division can create a cell that behaves differently forever, for better or worse. Red blood cells lose their nucleus, making more room for oxygen. Most cells keep a nucleus like a command center. Red blood cells give theirs up. During their development, they eject the nucleus and reorganize their interior so hemoglobin can fill the space. Hemoglobin is the oxygen-carrying protein that makes blood red, and packing more of it into each cell makes oxygen delivery more efficient. Losing the nucleus also helps red blood cells become unusually flexible. They can squeeze through capillaries that are narrower than the cell's resting diameter, then spring back into shape. The trade-off is real. Without a nucleus, red blood cells cannot repair DNA or make new proteins. They are built for a specific mission. Then they wear out and are replaced. This is specialization taken to an extreme. A red blood cell is less like a full city and more like a dedicated cargo ship. It carries a precious load through the body, and it is designed to do that job as well as possible. Neurons often last a lifetime, but their connections still change. A neuron can be long-lived, yet never truly fixed. The cell may remain, but the way it connects can shift with experience. When you learn a new skill, practice a language, or adapt to a new environment, neurons adjust the strength of their synapses. Some connections become more efficient, and some fade when they are rarely used. New branches can sprout, and old ones can be trimmed back. This constant adjustment is one reason memory feels physical. It is stored in patterns of connection and change, not in a single place you could point to. It is also why recovery can happen after certain injuries. The brain can sometimes reroute signals and recruit new pathways. These changes are not random. They are guided by activity and by chemical signals that reward useful circuits. Neurons are not constantly replaced in many parts of the brain, but the network they form is always being edited. Your life leaves traces in living wiring, and that wiring keeps reshaping itself. Skin cells rise upward, then die to form a protective layer. The surface of your skin is made of cells that are no longer alive in the usual sense. They begin deeper down, where new cells are formed. Then they migrate upward through layers, gradually filling with tough proteins and losing their internal machinery. By the time they reach the surface, they have become flat protective tiles. They lock together, help hold moisture in, and keep many microbes out. Then they flake away, replaced by the next wave rising from below. This is why your skin can be both strong and self-renewing. It is also why it can heal after minor injuries. The living layers beneath can produce new cells and rebuild the barrier. The idea can feel strange at first. Part of what you touch when you touch your own arm is the remains of former living cells. Yet that sacrifice is useful. It turns a vulnerable organism into something with armor that can renew itself, quietly and continuously. Bone constantly remodels, dissolving and rebuilding in living balance. Bone looks permanent, but it behaves more like a construction site that never closes. Tiny teams of cells remove old mineral and collagen, and other teams lay down fresh material in its place. This remodeling repairs microcracks that form during daily movement before those cracks become serious fractures. It also helps manage minerals in the bloodstream, especially calcium and phosphate. When the body needs more, bone can release it. When the body has enough, bone can store it. Remodeling changes across life. During growth, building dominates. With age or certain hormones, breakdown can outpace rebuilding, which can weaken the skeleton. Still, the core idea is beautiful. Your skeleton is not just a frame. It is a living reservoir and a living repair system. Every step you take creates tiny stresses. Your bones respond by renewing themselves from within, maintaining strength through constant change rather than rigid permanence. cancer begins when cell rules break and growth escapes control. In healthy tissue, growth is a privilege that must be earned. Cells wait for clear permission, they stop when space is tight, and they retire when their job is done. Cancer begins when that social contract unravels. A cell gains changes that tell it to keep dividing, to ignore stop signals, or to dodge the usual consequences. At first, the change may be invisible. One cell simply grows when it should not. Over time, it can recruit support from its surroundings, persuading nearby tissue to feed it and protect it. What makes this so unsettling is that the tools used are normal tools. Division, survival, and repair are all part of ordinary biology. Cancer is the misuse of those powers. It is what happens when a cell stops acting like a citizen of the body and starts acting like it is the whole body. Tumors evolve inside the body, competing for space and resources. A tumor is not a uniform lump. It is more like a shifting ecosystem. As cells divide, small differences appear between them. Some grow faster. Some tolerate low oxygen. Some survive immune pressure better. The environment inside a tumor is harsh because nutrients are limited and waste builds up. That harshness becomes a selection test. Cells that cope well leave more descendants, and the population changes over time. Treatments can add another filter. If most cells are sensitive to a drug, the rare survivors can expand afterward. This is evolution on a compressed timescale, happening inside one person. It is also why cancers can behave so differently from one another, even when they start in the same tissue. The tumor is shaped by competition and chance, and it keeps changing as conditions change. Understanding that dynamic is crucial because it explains both resistance and relapse. Cells have checkpoints and they can pause division to fix DNA. Cell division is not a reckless sprint. It is a process with stoplights. At key moments, cells check whether DNA has been copied correctly, whether chromosomes are properly attached, and whether damage is present. If a problem is detected, the cell can pause the cycle and bring repair tools to the site. This pause is protective because copying errors can become permanent if the cell divides too soon. Some checkpoints can even trigger a deeper response, where the cell chooses to enter a long stop rather than risk passing errors onward. These controls are especially important in tissues that renew often, because frequent division creates many opportunities for mistakes. When checkpoint systems fail, damaged cells can continue dividing, and that raises the chance of disease. It is comforting to know that your cells are not blindly reproducing. They are inspecting, waiting, correcting, and only then moving forward when conditions look safe. Cells repair DNA every day because damage is always happening. DNA is sturdy enough to store information for a lifetime, yet it is constantly being challenged. Heat, reactive chemicals, and ordinary metabolic activity can nick bases, snap strands, or create small distortions. Even normal copying during division can introduce misprints. Cells respond with repair crews that recognize specific types of damage and fix them using matching rules. Some systems clip out a faulty section and rebuild it. Some systems stitch broken strands back together. This work happens quietly, all day and all night, and most of it never affects you. When repair fails, the consequences can accumulate. Mutations can build over years, and the risk of malfunction rises. The amazing part is scale. Each cell is a tiny workshop performing maintenance on its most valuable archive without ever opening it to the outside world. Your health depends not only on what your DNA says, but on how well your cells keep that message intact. Sunlight can injure DNA, even before you feel a sunburn. The warmth of someone's skin feels harmless at first, but ultraviolet light carries enough energy to change molecules. When it hits DNA, it can cause neighboring bases to stick together in abnormal ways. That tiny chemical mistake can disrupt copying and reading of genes. You might not see anything immediately. Redness and pain come later, after cells respond with inflammation and repair. Meanwhile, your skin cells are already running damage control, pausing division, fixing lesions, and removing cells that are too compromised to trust. This is why sunscreen matters. It is not only about comfort or appearance. It is about reducing invisible molecular injury. It is also why tanning is not a sign of health. Pigment is a protective response to stress, not a reward for being outside. Sunlight is essential for life, but parts of it are also a hazard. Your cells treat UV exposure as an emergency they must manage fast. Telomeres protect chromosome ends, like caps that prevent fraying. Chromosomes have ends, and those ends face a tricky problem. The machinery that copies DNA struggles to fully replicate the very tips. Without protection, important genes could be shaved away over time, and chromosome ends could be mistaken for broken DNA that needs repair. Telomeres solve this by acting as sacrificial buffers. They are repeated sequences that can take the wear, so essential information stays safer. They also form structures that help hide the ends from repair systems that might fuse chromosomes together. In this way, telomeres protect both accuracy and order. The concept is simple, but the impact is huge. A chromosome is not only a string of genes. It is a physical object that must be maintained. Telomeres are like the finishing work on a book that keeps the pages from tearing loose. They remind us that information storage in biology is not only about code. It is also about protecting the edges of the code. Telomeres shorten with divisions, but some cells rebuild them. Each time many cells divide, telomeres tend to get a little shorter. That gradual loss acts like a counting system. After enough divisions, shortening can push a cell toward retirement, which helps limit uncontrolled growth. Yet some cells need to divide many times across a lifetime. Bud-forming cells, for example, must keep producing new cells steadily. To manage this, certain cells can rebuild telomeres using an enzyme called telomerase. Telomerase adds repeats back onto the ends, restoring buffer length and allowing more divisions. This ability is carefully controlled because unlimited telomere rebuilding can remove an important safety break. Many cancers reactivate telomerase for exactly that reason. The balance is delicate. Too little rebuilding can contribute to tissue decline. Too much can support runaway growth. When you hear about telomeres, it can sound like a simple clock, but it is more like a negotiated contract between renewal and restraint. Cells need both. Viruses hijack cells, turning them into virus-making factories. A virus is not a full living cell. It cannot make proteins on its own, and it cannot harvest energy. Instead, it enters a host cell and rewires it. Viral genetic material takes over the cell's machinery, directing ribosomes to build viral parts and redirecting membranes to assemble new virus particles. The host cell becomes a workshop for something that does not serve it. Some viruses shut down the cell's normal protein production, so the cell's resources are focused on the intruder's needs. Some hide their genetic material inside the nucleus, blending in with the cell's own processes. The result can be dramatic. A cell that once supported your tissue can become a launch site, releasing new viruses that spread infection onward. This is why antiviral defenses are so important. Your body is not only fighting particles in the bloodstream, it is fighting the takeover of its own cells. Viruses reveal a startling truth. Life's tools can be stolen and used against the life that built them. Some bacteria can live inside your cells, hidden from many defenses. Most bacteria are tackled outside cells, where immune proteins and roaming defenders can reach them. Some species take a different approach. They slip into your cells and use the interior as shelter. Inside, they can avoid certain antibodies and dodge parts of the immune system that patrol the bloodstream. Some hide within immune cells themselves, turning the very defenders into unwitting taxis. Others occupy membrane-bound compartments and manipulate the cell, so those compartments do not merge with digestive systems. This strategy can make infections harder to clear because the battleground is now inside your own tissue. It can also change symptoms. An infection can become chronic, with bacteria persisting in protected niches. The more you learn about this, the more impressive immune surveillance becomes. Your body is not only checking for outsiders in open space, it is scanning the insides of cells for signs that something foreign is living where it should not. Some bacteria have evolved to make that job much harder. Cells can recognize viruses, then trigger alarms to neighbors. When a virus enters a cell, it often leaves clues behind. Strange RNA forms appear, unusual proteins are made, and normal patterns are disrupted. Cells can detect these signs and respond fast. One key response is sending warning signals outward, including interferons that tell nearby cells to raise their defenses. Those neighboring cells may reduce viral entry, increase virus-destroying enzymes, and prepare to display viral fragments if infection occurs. This turns one cell's discovery into a neighborhood alert system. It also buys time for the immune system to mobilize specialized fighters. Some viruses try to silence these alarms because early warning is bad for viral spread. The back and forth is an arms race that has shaped both viruses and hosts for millions of years. The remarkable part is that a cell does not need to be conscious to act protectively. It can sense danger, broadcast it, and coordinate a wider response. Your tissues are not passive targets. They are early warning networks. Cells use receptors to detect hormones, like insulin, with specificity. A hormone drifting through blood is only meaningful to the right listener. Cells create that selectivity by displaying receptors, which are proteins shaped to recognize particular messenger molecules. When the match occurs, the receptor changes shape, and that change becomes an internal instruction. It is the reason one chemical signal can travel widely without affecting every tissue equally. Insulin is a classic example. It circulates broadly, yet only cells with the correct receptors respond by adjusting how they handle sugar and energy storage. This also explains why receptor numbers matter. A cell can become more sensitive by placing more receptors on its surface, or less sensitive by pulling receptors inward. Some medicines work by blocking receptors, and others mimic natural hormones by activating them. In that sense, cell biology is also about language. Receptors are the ears, and hormones are the words. One hormone can cause different effects depending on the cell type. A message is only half the story. The receiving cell decides what the message means. Two cells can bind the same hormone, then launch very different responses because their internal machinery is different. One cell may switch on genes that encourage growth. Another may change its membrane channels and alter electrical behavior. Another might release a second messenger that affects nearby tissue. This flexibility is how the body reuses a small set of chemical signals for many purposes. Adrenaline is a good illustration. In one tissue, it can increase heart rate. In another, it can influence how fuel is released into the blood. The hormone did not change. The interpretation changed. That interpretation depends on receptor subtype, on the proteins connected to it, and on which genes are available to activate. It is a reminder that cells are not passive receivers. They are active translators, turning the same signal into different actions across the body. Cells amplify signals, so a tiny cue can cause a big response. If cells required massive signals to act, life would be slow and wasteful. Instead, cells use amplification, where one small event triggers many larger events downstream. A single activated receptor can switch on enzymes that produce thousands of second messenger molecules. Those messengers can activate many targets, and each target can activate many more. The result is like a whisper that becomes a crowd response. This is how your eyes can respond to low light, and how immune cells can react rapidly to a small amount of danger signal. Amplification also allows fine control because the cascade can be damped or boosted at multiple steps. It is not only about strength. It is about shaping the response in time. Some cascades create sharp bursts. Others build slowly and last longer. When amplification goes wrong, signals can become too loud or too quiet. Many diseases are, at their core, problems of signal volume. Calcium ions act like quick signals, switching processes on and off. Calcium is a building material for bones, but inside cells it plays a second role as a fast signal. Cells keep calcium levels low in the cytoplasm, then briefly raise them when action is needed. That quick rise can trigger muscle contraction, secretion of neurotransmitters, or activation of enzymes that change cell behavior. Calcium can arrive through channels in the membrane, and it can also be released from internal stores like a sudden wave. Because the change is rapid, it works well for moments when timing matters. A pulse of calcium can mean start now. A different pattern can mean stay active longer. Cells even use calcium waves that travel across the cell like ripples, coordinating distant regions of the cytoplasm. This is chemistry used with the precision of a clock. The same ion that forms hard mineral in teeth can also be the brief spark that tells a cell to move, release, or respond. Cells use electrical charges, not just chemicals, to send information. Electricity sounds like wires and outlets, but cells can generate electrical patterns using simple ions. When charged particles move across a membrane, they change voltage. That voltage can influence which channels open next, and that becomes a form of information. Many cells use this, not only neurons. Heart cells coordinate their beat through electrical coupling. Certain cells in glands and epithelia use voltage to control secretion and fluid movement. Even a single cell can maintain an internal voltage difference, and that difference can guide transport of nutrients and ions. Electrical signals are fast, and they can travel further than many chemical signals inside crowded tissue. They also integrate well with chemistry, because voltage can control the flow of calcium and other ions that trigger chemical cascades. In living systems, electricity and chemistry are partners. Cells do not need metal wires to be electrical. They only need membranes and the right gates. Neurons fire by moving ions, creating fast electrical pulses. A neuron communicates by briefly flipping its voltage, and that flip travels along the cell like a wave. It begins when certain channels open and let ions rush in. That change triggers nearby channels to open in sequence, so the signal moves forward without fading. Afterward, other channels help reset the membrane to its resting state, ready for the next pulse. This is why nerve signals can travel quickly across long distances, from a fingertip to the spinal cord, or from the ear to the brainstem. The pulse itself is not a thought. It is a reliable courier that carries timing. The meaning comes from patterns, which neurons interpret through their connections. Some neurons fire in bursts. Others fire steadily. Some are silent until a specific trigger arrives. The beauty is that this system works in the messy environment of wet tissue, and it works with remarkable consistency. Your ability to react, sense, and move relies on ion traffic guided by protein gates. Synapses change strength, shaping learning at the cell level. Learning leaves traces in connection strength. At a synapse, one neuron influences another by releasing chemical messengers into a tiny gap. The receiving side can respond more strongly or more weakly over time, depending on patterns of activity. When two neurons often activate together, the connection can become easier to trigger. That can involve adding more receptors, releasing more messenger, or changing the structure of the synapse itself. Other connections weaken when they are rarely used, which helps the brain avoid overload and refine what matters. This is one reason practice works. Repetition does not just add knowledge. It reshapes the probability of signals traveling along certain routes. It is also why memories can be fragile. Connections can shift with sleep, stress, and new learning. The brain is not storing information like files in a cabinet. It is storing tendencies in living circuitry. Each strengthened synapse is a small vote for a pathway to be used again. Cells can remember past stress, then respond faster next time. A cell that has being challenged can become quicker to defend itself later, even if the original threat is gone. This kind of memory is not conscious, and it is not the same as immune memory in specialized cells. It is more like readiness. After heat stress, a cell may keep higher levels of protective chaperone proteins that help refold damaged molecules. After exposure to a toxin, it may maintain stronger detox systems or faster transporters that remove harmful compounds. In plants, repeated drought can prime cells to close pores more rapidly when water begins to drop again. In microbes, earlier stress can tune gene networks, so a later shock causes a faster shift in metabolism. This phenomenon is often called cellular priming. It shows that experience can shape response even in single cells. The past can linger as altered protein levels, altered signaling sensitivity, and altered gene accessibility. A cell is not only reacting in the moment, it is carrying a history. DNA stays mostly still, while messages copy out and travel. It is tempting to imagine DNA being carried around like a manual, but cells rarely move the original instructions. Instead, they make working copies that can travel. That choice protects the most valuable information from daily chaos. DNA is large, delicate, and deeply entangled with proteins that help organize it. Moving it would be risky and inefficient. So the cell keeps the master copy sheltered, then sends out short messages to the places where building happens. Those messages can be made quickly, used briefly, and destroyed when they are no longer needed. This lets a cell react fast, without exposing its core archive. It also allows fine control. A cell can copy only the sections it needs, and it can adjust how many copies it makes. Stability stays at the center, and flexibility moves outward. That balance is one reason life can be both durable and responsive. Messenger RNA is a temporary note, not the whole instruction book. Messenger RNA is designed to be disposable. That sounds harsh, but it is one of the reasons cells can stay safe and adaptable. A short-lived message limits how long a set of instructions can be used. If a signal changes, the cell can stop making that message, and the effect fades naturally as the old copies break down. This prevents outdated orders from lingering. It also reduces risk. If a message is damaged, it does not corrupt the DNA itself. The cell can simply make a fresh copy. Different messages can also be handled differently. Some are kept around for longer, and some are destroyed almost immediately. That lifespan becomes a powerful dial for control. It is like writing on a sticky note instead of carving into stone. The note can be edited, replaced, or removed, and the master archive remains protected. Ribosomes build proteins by snapping amino acids into exact order. A ribosome is small enough to be invisible to any normal microscope, yet it performs one of life's most precise assembly jobs. It reads a message step by step, and for each step it selects the next amino acid from a crowded pool. The match has to be correct because a protein is not just a chain. It is a chain that must fold into a working shape. The ribosome helps keep the pace steady, and it checks the fit as each amino acid arrives. When it finishes, a brand new protein emerges, ready to become a sensor, a scaffold, or an enzyme that speeds up reactions. Some proteins are built in the open interior of the cell. Others are built on ribosomes attached to membranes, so the new protein can be threaded directly into the right compartment. This is manufacturing at the molecular scale, and it happens constantly in every living cell. The endoplasmic reticulum folds proteins, like careful quality control. Making a protein is only the beginning. Many proteins must be folded into a precise shape, and the endoplasmic reticulum is where much of that careful work happens. Inside its winding spaces, helper molecules guide new proteins as they twist and settle. If a protein misfolds, it can fail at its job or cause harm. So the cell checks, corrects, and sometimes rejects. This matters in dramatic ways. Some diseases involve proteins that fold poorly, which can lead to cellular stress and loss of function. The endoplasmic reticulum also helps prepare certain proteins for life outside the cell by adding chemical tags that act like labels. It is not a passive hallway. It is a workshop with standards. When the workload becomes too heavy, the cell can trigger an internal response that slows production and boosts support systems. It is a reminder that biology is not just building. It is building correctly. The Golgi apparatus packages proteins, then ships them to locations. Once a protein is ready, it still needs a destination. The Golgi apparatus is the cell's shipping center, where molecules are sorted, labeled, and sent out in tiny membrane bubbles. A protein meant for export is wrapped and directed toward the cell surface. Another protein might be routed to a digestive compartment. Another might be sent to a specific region of the membrane, so it can act as a receptor. This sorting depends on chemical tags that function like addresses. The Golgi can also modify proteins as they pass through, adding sugar chains that change how they behave and how long they last. It is easy to miss how remarkable this is. Inside a space smaller than a speck of dust, there is a delivery system that moves cargo to exact targets. When this system fails, cells can lose organization and tissues can suffer. Order depends on proper delivery. Lysosomes break down waste using powerful chemical tools. Every cell generates trash, and some of that trash is dangerous if it lingers. Lysosomes are the cleanup crew, filled with enzymes that can dismantle worn-out parts into reusable pieces. They work best in an acidic environment, which keeps their tools effective and helps protect the rest of the cell. If those enzymes leaked into the cytoplasm, they could cause damage. So the lysosome keeps its harsh chemistry contained. Lysosomes also help defend the body. In certain immune cells, they can digest engulfed bacteria. They can also clear out broken organelles and help recycle material when nutrients are limited. When lysosomes malfunction, waste can build up and cells can struggle to survive, especially in long-lived tissues like the brain. It is a sobering idea. Health depends not only on making new things, but also on removing old things safely. Lysosomes make that possible. Peroxisome neutralize toxic molecules before they spread damage. Some chemical reactions create byproducts that are useful in small amounts but harmful in large amounts. Peroxisomes handle some of the most reactive of these, including forms of oxygen that can damage proteins and membranes. They contain enzymes that convert these molecules into safer substances, often turning a potential threat into something the cell can reuse. Peroxisms also help break down certain fats that are difficult for other compartments to process. That matters in organs like the liver, where fats and toxins are constantly being managed. When peroxisms do not work correctly, the effects can be severe because toxic compounds can accumulate and disrupt development or tissue function. What makes this so fascinating is the strategy. Instead of letting reactive chemistry roam free, the cell isolates it in a dedicated space with the right tools. It is hazard management at a microscopic scale. Peroxisms prove that cells are not just alive, they are careful. The cytoskeleton gives cells shape, strength, and moving parts. A cell is not a simple blob. Inside, there is an internal framework that can hold a shape, resist pressure, and rearrange itself when the cell needs to change. That framework is the cytoskeleton. It includes different kinds of fibers. Some are rigid for support, and some are flexible for tension. With this system, a cell can spread out, pinch inward, or form extensions to explore its surroundings. During division, this internal scaffolding reorganizes dramatically to help separate copied material into two new cells. In nerve cells, the cytoskeleton supports long extensions that can stretch astonishing distances in the body. It is also a stage for motion. Proteins can pull on these fibers like tiny muscles, shifting parts of the cell and helping it respond to signals. The cytoskeleton is why cells can be both sturdy and adaptable. It is structure that can move. Microtubules act like tracks, carrying cargo across the cell. Microtubules are one of the cell's most impressive inventions. They form long, hollow tubes that reach across the interior, creating routes for transport. On these routes, motor proteins can walk while carrying cargo. Some motors move toward one end of the microtubule, and others move toward the opposite end. That polarity turns the cell into a map with directions. Cargo can include food-filled vesicles, signaling molecules, and materials needed to build new structures. In neurons, microtubule transport becomes especially dramatic because supplies must travel down long nerve extensions to reach distant synapses. If transport breaks down, parts of the cell can starve even when resources exist elsewhere. Microtubules also play a starring role in cell division, where they form a spindle that helps separate chromosomes. The same system that hauls packages can also organize inheritance. It is logistics and destiny, built from protein. Epigenetic marks tune genes without changing the DNA letters. DNA provides the letters, but cells also need punctuation and emphasis. Epigenetic marks help provide that control. They are chemical tags added to DNA or to the proteins that package it, and those tags influence whether a gene is easy to read or difficult to access. This allows a cell to maintain identity over time. A muscle cell keeps acting like muscle because gene access patterns stay stable. It also allows flexibility. Environment can influence epigenetic marks, which can shift gene activity without altering the sequence itself. During development, epigenetic changes help guide the transition from one cell state to another, step by step. Some marks are long-lasting, and some are reversible. That mix supports both stability and adaptation. Epigenetics is also one reason identical twins can diverge in traits over life, even with very similar DNA. The code can be the same, yet the way it is read can differ. Two cells can share cytoplasm, briefly merging their inner worlds. Cells are usually separated by membranes, yet biology has ways to blur that boundary. Some cells form thin bridges that allow bits of cytoplasm to pass between them. Others fuse fully, creating one larger cell with multiple nuclei. Muscle fibers form this way, which helps them act as powerful, unified units. Certain immune cells can fuse into giant cells when dealing with persistent threats too large for a single cell to handle. There are also cases where developing cells share cytoplasm through temporary connections, which can help coordinate growth and distribute materials. This sharing changes what a cell can do. It can spread signals quickly, share resources, and synchronize behavior across a group. It also raises fascinating questions about individuality at the cellular level. Where does one cell end and another begin, if they can merge and cooperate so directly? The body is built from boundaries, yet it also depends on carefully chosen exceptions to those boundaries. Cells use mitochondria, and those mitochondria carry their own DNA. Inside many of your cells are tiny energy makers with a surprising secret. They keep a small genetic record of their own, separate from the DNA in the nucleus. That extra DNA helps encode some of the tools needed for energy production, and it hints at an ancient independence. It also means mitochondria are not just passive parts. They have their own lineage, their own copying process, and their own vulnerabilities. Because mitochondrial DNA is close to the chemical reactions that generate energy, it can face more wear over time. Changes there can affect tissues that need steady power, like muscles and nerves. This is also why family history can matter in unusual ways. Mitochondria are typically inherited through the egg, so this genetic thread often follows the maternal line. In one cell, you can find two genetic stories running in parallel, and they cooperate to keep you alive. Mitochondria likely began as bacteria that moved in long ago. Long before animals existed, a strange partnership may have changed life forever. A larger cell likely took in a bacterium that was good at harvesting energy, and instead of digesting it, the cell kept it. Over time, the guest became a permanent resident. The resident provided efficient energy production. The host provided protection and steady resources. This idea is called endosymbiosis, and it explains why mitochondria still resemble bacteria in key ways. They divide in a bacteria-like fashion. They have their own DNA. Their inner membranes look like something built for chemical power generation. Over many generations, the partnership became so deep that neither side could easily return to life alone. The result was a new kind of cell, one with far more energy to spend. That extra energy may have made complex life possible, from larger bodies to faster brains. Your cells carry the echo of an ancient merger that rewrote what life could become. Chloroplasts in plants also began as captured bacteria long ago. Plants run on sunlight, but the machinery that captures that light may have started as an independent bacterium. In this scenario, a cell took in a photosynthetic bacterium and kept it, turning it into a chloroplast. Like mitochondria, chloroplasts retain their own DNA, and they divide within the cell rather than being built from scratch each time. The payoff was enormous. With chloroplasts, cells could convert light energy into chemical energy and build sugars from carbon dioxide. That ability reshaped Earth. Over deep time, photosynthesis helped change the atmosphere and made new ecosystems possible. It also created the base of most food chains we depend on. When you look at a leaf, you are seeing countless chloroplasts working in parallel, each one like a tiny solar panel with a bacterial past. A whole forest is, in part, the result of ancient cellular adoption. Plant cells build cellulose walls, making sturdy living architecture. Unlike animal cells, many plant cells live inside firm walls that are built outside the membrane. These walls are made largely of cellulose, a tough chain of sugar molecules that can form strong fibers. The wall gives plant cells shape and support, which is why plants can stand upright without bones. It also helps protect against physical damage and keeps cells from bursting when water flows in. The fascinating part is how this strength still allows growth. Plant cells loosen parts of the wall, then add new material in carefully controlled directions. That is how stems elongate and roots push through soil. Walls can also be reinforced to form wood, creating long-lived structures that can outlast generations. Every piece of timber, every paper page, and every cotton thread traces back to this cellular strategy. Plant architecture begins at the microscopic scale, where a cell wall turns soft living tissue into something that can hold up a whole tree. Plant cells use vacuoles to store water, salts, and chemicals. Many plant cells contain a large internal reservoir called a vacuole, and it can take up most of the cell's volume. This reservoir stores water and dissolved salts, and it helps create internal pressure that keeps leaves firm and stems upright. When a plant wilts, it is often losing that pressure. Vacuoles also store useful and sometimes intense chemicals. Pigments that color flowers can be held there. Defensive compounds that discourage insects can be tucked away safely. Even waste products can be stored in a way that keeps them from interfering with daily chemistry. In some fruits, vacuoles help set flavor by holding acids and sugars in balance. What feels like simple greenery is supported by hidden storage systems that act like water towers and chemical vaults. A plant does not only rely on roots and leaves. It relies on millions of cellular reservoirs managing pressure, protection, and supply. Plants move substances cell to cell through microscopic channels. A plant may look like a collection of separate cells, but many plant cells are connected by tiny channels that cross the cell walls. These channels allow small molecules and signals to pass directly from one cell to the next. It is a kind of shared network that helps tissues coordinate. Sugars made in a leaf can begin their journey toward roots and fruits. Signals about injury can spread, preparing nearby regions to respond. Developmental cues can travel, helping cells decide what to become as a bud forms or a root tip grows. These channels must be carefully controlled. Too open, and pathogens could spread more easily. Too closed, and the plant would lose coordination. Some plants can tighten these channels in response to threat, like closing doors during an emergency. The result is a living system that can act as a whole without a nervous system. Information and materials move through direct cellular connections, stitching plant life into one continuous body. Fungi cells build walls from chitin, more like insects than plants. Fungi are often confused with plants, but their cell walls reveal a different identity. Many fungi build walls from chitin, a tough material also found in insect exoskeletons. This gives fungal cells strength and protection, and it helps them grow in a distinctive way. Instead of building tall structures with rigid stems, many fungi extend thread-like tubes called hyphae. The wall at the tip is remodeled as the hypha pushes forward, exploring soil, wood, or living tissue. Chitin helps that growing tip stay durable while still flexible enough to expand. This is part of why fungi are such powerful decomposers. They can penetrate tiny cracks in dead wood and spread through complex environments. It also helps explain why fungi occupy their own kingdom of life, not plant and not animal. Their walls are engineered for a lifestyle of exploration and absorption, and chitin is a key piece of that design. Yeast cells can switch lifestyles from single to clustered communities. Yeast might seem like simple single cells, yet they can behave socially when conditions call for it. In rich environments, Many yeast live as individual cells that grow and divide. Under stress, some species can shift form, sticking together, or growing into elongated shapes that act more like a community. Clustering can help them share resources, resist harsh chemicals, and survive drying conditions. It can also change how they interact with surfaces, which matters in both nature and industry. In fermentation, yeast populations can organize in ways that affect how quickly sugars are converted and what flavors develop. In the wild, switching lifestyles can help yeast colonize fruit skins, tree bark, or soil pockets that change with seasons. The deeper lesson is that even a single-celled organism can have options. Life is not locked into one way of being. With the right triggers, yeast can shift from solitary growth to collective survival. Bacteria can trade genes, sharing useful tricks between neighbors. Bacteria do not rely only on slow genetic change across generations. They can share genes directly, and that can spread new abilities with startling speed. Some bacteria pass small DNA circles called plasmids from one cell to another through direct contact. Others can pick up free DNA from the environment, using it like a toolkit found on the ground. Some even exchange genes through viruses that move between cells. This gene trading can spread traits like toxin resistance, new metabolic pathways, or the ability to cling to surfaces. It is one reason antibiotic resistance can appear and spread so quickly in bacterial populations. From the bacteria's perspective, it is efficient. If a neighbor has solved a problem, why wait to evolve the same solution? Borrow it. This changes how we think about evolution. In bacteria, innovation can move sideways through a community, not only downward from parent to offspring. A microbial neighborhood can act like a shared library of survival strategies. Some bacteria form spores, surviving harsh conditions for years. When conditions become brutal, some bacteria switch into a survival form called a spore. In this state, the cell locks down its vital components, dehydrates itself, and builds protective layers that shield it from heat, drying, chemicals, and radiation. The spore is not growing or feeding. It is waiting. When water and nutrients return, it can reactivate and become a normal cell again. This ability explains how certain bacteria persist in soil, dust, and even on surfaces for very long stretches of time. It is also why sterilization can be challenging. Spores are built to endure. From an evolutionary perspective, spores are a time machine. They allow a lineage to bridge disasters that would otherwise wipe it out. A spore can outlast seasons, droughts, or extreme cold, and then resume life when the world becomes welcoming again. Survival is not always about fighting in the moment. Sometimes it is about becoming patient. Bacteria have no nucleus, yet their gene control can be elegant. Bacteria do not keep their DNA behind a nuclear wall, yet they manage it with impressive finesse. Many can switch whole sets of genes on or off in response to a single change in the environment. If a new sugar appears, some bacteria activate the enzymes to digest it and shut them down again when it is gone. If oxygen drops, they can shift metabolic pathways to keep producing energy. This control often relies on DNA-binding proteins that sit near genes like tiny switches, blocking or allowing transcription. There is also a clever economy to it. Genes that work together are often grouped, so one regulatory decision can coordinate an entire pathway. It is fast, efficient, and tuned to survival. Bacteria may be small, but they run sophisticated decision systems. They sense, compute, and respond in minutes, using gene regulation as their main language of adaptation. Some cells thrive in boiling vents, proving life can bend rules. Deep in the ocean, Hydrothermal vents pour out water hot enough to scald, and yet life gathers there. The key players are heat-loving microbes that treat chemical energy as their fuel. Instead of relying on sunlight, they use reactions involving compounds released from the vent to power growth. To survive the heat, their proteins are built to stay folded and stable at temperatures that would unravel most biology. Their membranes are also engineered for extremes, keeping the cell's boundary intact when heat tries to loosen everything. Even their genetic material is protected by specialized proteins that help it resist damage. These organisms do more than endure. They form the foundation of entire ecosystems, supporting worms, crabs, and other creatures that live in darkness. Vent life expands the definition of habitable. It suggests that where there is energy and chemistry, life can invent ways to persist, even in places that seem impossible. The smallest cells are near the size limit for life. There is a minimum size below which a cell cannot do the basic tasks of living. It needs room for genetic instructions, for ribosomes to build proteins, for membranes to maintain gradients, and for enough chemistry to keep reactions running. Shrink too far and you lose capacity, because molecules start crowding each other and essential components will not fit. Some of the smallest known bacteria are only a fraction of a micrometer across, and they live with extreme efficiency. Many rely on nearby organisms, borrowing nutrients and sometimes depending on a host for key molecules they no longer make themselves. This reveals a deep truth about life. Being small is not automatically simple. A tiny cell is often a masterpiece of compression, using the minimum toolkit that still allows growth and replication. Studying these minimal cells helps scientists ask a haunting question. What is the smallest package that can still be alive? Viruses are not cells, yet they shape the evolution of cells. Viruses do not have the full machinery of life, but they are powerful evolutionary forces. They infect cells, copy themselves, and apply pressure that rewards better defenses. Over time, cells evolve new ways to block entry, detect viral genetic material, and cut it apart. Viruses counter with new tricks, and the cycle continues. This arms race can drive rapid change in immune genes and cell surface proteins. Viruses also move genetic material around. When they infect, they can sometimes carry fragments from one host to another, creating new combinations that would be unlikely otherwise. Even long after an infection, traces can remain. In many animals, pieces of ancient viral DNA are embedded in genomes, like fossils written into heredity. Some of those sequences have been repurposed by evolution for new functions. In that way, viruses are both threats and unexpected contributors. They are not alive in the same way cells are, yet they have helped sculpt cellular life for eons. Endosymbiosis helped complex cells arise by joining forces. Complex cells did not only become complex by adding more genes, they also became complex by forming partnerships. Endosymbiosis describes a leap where one organism lived inside another in a stable relationship, and the two became inseparable. Over time, many genes that were once carried by the internal partner moved to the host's genome, which tightened the bond and reduced redundancy. The internal resident kept only the genes that were best held locally for its specialized work. This gene sharing created a system where the host provided protection and resources, and the resident provided a powerful new capability. The result was not simply cooperation. It was integration. This kind of merger helps explain why mitochondria and chloroplasts have double membranes and their own genetic remnants. It also shows how evolution can innovate by combination, not only by mutation. Life sometimes advances by learning to live together so closely that two lineages become one. Multicellular life began when cells cooperated and specialized. Single cells can do many tasks, but they face limits. Multicellularity became possible when groups of cells stayed together and began to divide labor. Some cells focused on movement. Some focused on digestion. Some focused on reproduction. Specialization allowed a larger organism to do more than any one cell could manage alone. Cooperation also required new rules. Cells had to coordinate growth, share resources, and prevent cheaters from taking more than they give. That is one reason cancer is such a deep problem. It echoes the ancient challenge of keeping a community stable. Multicellular life also required communication systems and adhesion systems, so cells could stay connected without losing flexibility. Over evolutionary time, this cooperation opened the door to nerves, muscles, immune systems, and complex development. Every animal and plant is a living alliance, built from cells that agreed through natural selection to limit their independence in exchange for becoming part of something larger. Cell adhesion proteins let tissues hold together, yet stay flexible. Tissues are not glued together by chance. Cells use adhesion proteins that act like molecular fasteners, linking cell to cell and cell to the surrounding matrix. These connections must be strong enough to survive pulling and stretching, and they must also be adjustable, because bodies move and grow. In skin, adhesion helps create a tough barrier that resists friction. In the heart, connections help cells contract as a coordinated sheet. In the immune system, adhesion can be temporary. A roaming immune cell can latch onto a blood vessel wall, roll along it, then squeeze through into tissue. Adhesion also carries information. When a cell attaches, it can trigger internal signals that influence survival, shape, and gene activity. Loss of proper adhesion can be dangerous because it can allow cells to detach and migrate in ways that contribute to metastasis. Adhesion proteins are therefore structural and communicative at the same time. They help tissues behave like cohesive materials without becoming rigid. During development, cells follow gradients, like maps made of chemistry. In an embryo, cells must know where they are and what they should become. One way they do this is by reading gradients, which are smooth changes in the concentration of signaling molecules across space. A cell in one region detects a high level of a signal and takes one path. A cell farther away detects a lower level and takes another. This is how broad patterns form without a central planner. The same principle can create a head-to-tail axis, shape a limb, or define layers in early tissue. Cells interpret gradients using receptors and gene networks that translate concentration into identity. Timing matters too, because the embryo is changing while signals are spreading. Gradients can also interact, creating a coordinate system where two signals together provide a more precise location than either alone. It is awe-inspiring to consider. An embryo builds its geometry using invisible chemical landscapes, and cells navigate them as if they were reading a map. One embryo becomes many tissues because cells choose different genes. Early in development, many cells share the same DNA, yet they begin to diverge. The difference comes from gene expression. Certain genes are activated in one cell, while others remain silent. These choices are influenced by signals from neighbors, by position in the embryo, and by internal regulatory networks that stabilize a fate once it begins. Over time, these decisions become layered. A cell does not jump straight from generic to fully specialized. It moves through stages, narrowing its options, committing to a lineage, and refining its role. This is how one embryo produces skin, muscle, bone, blood, and brain. The transformation is not a change in the genetic alphabet. It is a change in which pages are read, and how strongly they are read. This is why development can be both robust and fragile. A well-timed signal can guide cells toward healthy organization. A disrupted signal can send cells down the wrong path, with lifelong consequences. Cells can migrate long distances, sculpting organs before birth. Some of the most important construction work in an embryo involves cells that travel. They move in streams, in sheets, and sometimes as individual explorers. Their destinations matter. Cells that will form parts of the face migrate and shape the skull and jaw. Cells that will form the heart move into position and assemble into a structure that will soon beat. Nerve-related cells travel to populate the gut and help control digestion later in life. These migrations are guided by attraction cues, repulsion cues, and the texture of the surrounding matrix. Cells also stop when they arrive, and they anchor into new neighborhoods, changing shape and identity as they settle. When migration goes wrong, organs can form with gaps, misplacements, or missing cell types. When it goes right, it is astonishing. A body is not only grown where it starts. It is assembled by moving parts that know where to go, long before you take your first breath. Your immune system learns because certain cells keep memory. After an infection, your body does not simply return to a blank slate. Some immune cells keep a record of what they encountered, and they linger for years, sometimes decades. Next time the same threat appears, those memory cells can respond faster than a first-time reaction. They divide quickly, call in help, and produce a stronger wave of defense before a virus or bacterium gains much ground. This is also why vaccines work. They give the immune system a safe rehearsal, so the next encounter feels familiar. What is especially fascinating is that memory is not stored in one place. It is distributed across different cell types, each with its own role. Some keep watch in blood and lymph. Some settle in tissues, closer to common entry points. In a real sense, your immune system has a past, and it uses that past to protect your future. Antibodies are proteins, custom-shaped to fit specific targets. Antibodies are like tiny tools built for recognition. Each one has a shape that can bind to a particular feature on a microbe, almost like a key fitting a lock. Once bound, an antibody can block an invader from attaching to cells. It can also mark the invader for destruction, so other immune cells know exactly what to grab. What makes this even more mind-blowing is how many different shapes the body can generate. Immune cells shuffle genetic segments to create enormous variety, then refine that variety through selection. Cells that bind well are encouraged to multiply. Cells that bind poorly fade away. Over time, antibodies can become better at their job, which is why later responses can be stronger and more precise. This is protein engineering happening inside you, guided by biology rather than blueprints. A clear shape meets a clear target, and an invisible battle shifts in your favor. White blood cells squeeze through vessel walls, chasing infection. When infection flares in a tissue, immune cells in the bloodstream have to leave the highway and enter the streets. They do this through a carefully controlled process. First, they slow down, rolling along the vessel wall, as if scanning for the correct exit. Then they latch on tightly, using adhesion molecules like temporary climbing grips. After that comes the astonishing part. The cell flattens, reshapes, and squeezes between the cells of the vessel lining without ripping the vessel open. It is more like slipping through a zipper than breaking through a wall. Once outside, the immune cell follows chemical cues toward the trouble spot, moving through dense spaces that would stop most cells. This is why swelling and redness appear. The area is being flooded with defenders. The next time you notice inflammation around a cut, remember that cells are performing microscopic escapes and rescues. one squeeze at a time. Inflammation is coordinated cell behavior, not just swelling. Inflammation can look like a simple symptom, but it is a full-body strategy written in cell language. Blood vessels widen to increase flow, which brings heat and redness. Vessel walls become more permissive, allowing immune cells and proteins to enter the tissue. Local cells release signals that change the behavior of nearby cells, so the response is not random. Some immune cells attack and engulf invaders. Others release molecules that guide the battle and shape the cleanup. As the threat is controlled, different signals rise that promote repair and calm the reaction down. When this resolution step fails, inflammation can become chronic and damaging. That is when a protective response turns into wear on the body. The fascinating part is that inflammation is not a single thing. It is a sequence, like an emergency protocol. Cells coordinate roles, timing, and intensity to solve a problem, then return the tissue toward balance. Cells can present protein fragments, showing what they contain inside. Most infections happen inside cells, which creates a challenge for the immune system. How can defenders know what is happening behind a membrane? One solution is presentation. Cells chop proteins into small fragments and display them on their surface using special carrier molecules. This is like posting a sample from the inside on an outer bulletin board. If the fragments look normal, immune patrols move on. If the fragments look viral or otherwise suspicious, immune cells can target that cell for removal. This system also helps build immune memory because it teaches the body what to recognize. Some pathogens try to interfere with presentation because hiding their fragments buys time. What makes this process so gripping is its honesty. Cells are required to show what they are making, even when they would rather stay private. A healthy body depends on this transparency. It is surveillance based on evidence, carried out by proteins no larger than dust. Autoimmune disease happens when recognition mistakes self for threat. The immune system is trained to tolerate the body it protects, yet this training is not perfect. Autoimmune disease can arise when immune cells react strongly to the body's own molecules, treating them as if they belong to an invader. The consequences depend on the target. If immune activity focuses on joint tissues, movement can become painful. If it focuses on insulin-producing cells, blood sugar control can fail. In some cases, the trigger may involve infection, where similarities between microbial molecules and self molecules confuse the system. In other cases, genetics and regulation play a bigger role. What is striking is that the immune system is not malicious. It is powerful, and it is trying to do its job. Autoimmunity is a misfire of that power. It shows how difficult the problem of identity truly is at the cellular level. The body must defend itself without becoming its own enemy, and that balance is delicate. Some cells hide from immunity by turning down visible identity tags. Immune patrol depends on recognition, and recognition depends on what cells display. Some cells can evade detection by reducing the surface markers that immune cells use for inspection. This strategy appears in certain cancers and in some viral infections. If a cell presents fewer identifying molecules, it may be harder for immune cells to confirm that something is wrong inside. Yet this hiding act can come with risk, because other immune cells are tuned to notice missing signals as a sign of trouble. This creates a tense game of concealment and exposure. Tumor cells may shift their display over time, adapting to immune pressure like an animal changing camouflage. Viruses may block surface trafficking so infected cells show fewer warnings. The result is not a simple chase. It is a negotiation at the membrane, where survival can depend on what is shown and what is withheld. Cell biology becomes strategy, and the battlefield is visibility itself. Gut microbes influence your cells, including immune and brain signaling. Your gut is home to vast microbial communities, and they are not silent passengers. They break down food components you cannot digest alone, producing small molecules that your cells can absorb and respond to. Some of those molecules influence immune balance, helping tune how strongly the body reacts to everyday stimuli. Others interact with gut nerves and chemical pathways that can affect mood and stress responses through the gut-brain connection. Microbes also compete with pathogens, taking up space and resources that invaders might need. When the microbial community shifts, the signals it produces can shift too, and that can change how gut-lining cells behave and how immune cells respond. What is fascinating is that this influence is not mystical. It is chemistry and contact. Microbes release molecules. Your cells read those molecules. The relationship becomes a dialogue, and health depends on how well that dialogue is maintained. Cells respond to circadian clocks, changing chemistry by time of day. Time is built into biology. Many cells run internal clocks that help coordinate activity with day and night. These clocks influence which genes are active, which enzymes are abundant, and how energy is managed across hours. In the liver, timing affects how nutrients are processed. In immune cells, timing can shape how strongly responses rise. In the brain, timing supports sleep and wake patterns that influence memory and attention. These rhythms are guided by core clock genes that interact in feedback loops, creating cycles that reset each day. Light exposure, meals, and activity can nudge those cycles, which is why irregular schedules can feel disruptive. The most striking idea is that your body is not doing the same chemistry at all times. The same cell can behave differently at morning and at night, not because it changed identity, but because it is keeping time. Cell biology is not only about structure, it is also about rhythm. Even single cells can keep time using molecular rhythms. Timing does not require a brain. Even single cells can generate repeating cycles through networks of molecules that rise and fall in predictable patterns. Some of these rhythms are driven by feedback loops, where one protein promotes a process that later suppresses itself. Others rely on cycles of chemical modification, like adding and removing tags that change protein activity. In microbes, timing can help coordinate division and metabolism with environmental cycles. In isolated cells from larger organisms, clock-like rhythms can persist outside the body for days, which shows that the timing machinery is truly internal. What makes this awe-inspiring is that it is a form of order emerging from chemistry. Molecules interact, reactions loop, and time appears as a pattern. It is not a ticking sound. It is a repeating rise and fall in concentration and activity. When you zoom in far enough, life is not only responding to the world, it is keeping its own beat. Cells make heat, and brown fat cells specialize in warming you. Unlike most fat, brown fat is built for burning, not storing. Its cells are packed with mitochondria, and those mitochondria can release energy as heat instead of saving it as fuel. This is especially important for newborns, who cannot shiver well and lose heat quickly. In adults, brown fat can still switch on in cold conditions, helping defend body temperature without movement. Blood flow through these tissues increases, spreading warmth through the body like a built-in radiator. Scientists also study brown fat because it changes how the body uses calories, and it may influence metabolic health. What makes it so captivating is that warmth can be a cellular job. Heat is not only a byproduct of life. In some cells, heat is the purpose. Fat cells store energy, but they also send hormone signals. Fat tissue is not a silent warehouse. It is an active organ that speaks to the rest of the body through hormones and signaling molecules. These signals help regulate appetite, influence how sensitive cells are to insulin, and shape inflammation levels throughout the body. When fat cells expand, their signaling can change, and that can shift how the liver handles sugar and how muscles use fuel. Fat tissue also communicates with the brain, helping tune hunger and satiety across the day. This means body weight is not only about stored calories. It is also about messages that affect behavior and metabolism. The same tissue that saves energy for a future winter can also influence the feeling of fullness after dinner. Seeing fat cells as communicators changes the story. They are not just storage. They are participants in a constant conversation about energy, stress, and balance. Muscle cells fuse into long fibers, forming powerful living cables. Most cells stay separate, but many skeletal muscle cells merge during development, creating long fibers with many nuclei. This fusion allows a single muscle fiber to grow large and produce huge amounts of the proteins needed for contraction. It also helps the fiber repair. Special helper cells can add new nuclei to a damaged fiber, supporting rebuilding after strain. Inside each fiber are repeating structures that slide past one another to generate force, and the alignment of those structures depends on the fiber being one continuous unit. This is why muscles can pull with such strength and such control. A muscle is not just a bundle of cells working side by side. It is a bundle of cellular unions, built to act as living cables that can tighten, relax, and respond in milliseconds. Fusion turns many small builders into one powerful machine. Heart muscle cells beat in sync by passing signals through junctions. Your heart does not rely on one cell shouting orders to the rest. Heart muscle cells are linked by special junctions that let electrical signals pass directly from cell to cell. When one region becomes excited, the voltage change spreads through the network, coordinating contraction like a wave moving through a stadium crowd. This coupling helps the heart act as one pump instead of a patchwork of competing beats. The timing is shaped by specialized pacemaker cells and by pathways that conduct signals efficiently through the heart structure. When these connections are disrupted, rhythms can become chaotic and pumping becomes less effective. It is astonishing that a heartbeat is a community event at the cellular level. Each cell contributes, and each cell listens. The steadiness you feel in your pulse is the result of millions of cells sharing signals through microscopic doorways, beat after beat, all day long. Blood clotting is a cell-guided cascade, fast and coordinated. A cut is more than a leak. It is a call to action. Damaged tissue exposes signals that tell platelets to stick, pile up, and begin forming a plug. At the same time, a chain of clotting proteins activates in sequence, each step triggering the next, until a mesh of fibrin forms to stabilize the patch. The cascade is designed for speed because blood pressure does not wait. It is also designed for control because clots in the wrong place can be dangerous. Cells lining blood vessels help keep clotting quiet when vessels are intact, and they help trigger clotting when injury occurs. Once repair is underway, other systems dissolve the clot so flow can return to normal. What looks like a simple scab is actually an emergency protocol, coordinated between cells and proteins with remarkable timing. Platelets are cell fragments, but they act like emergency repair kits. Platelets are not full cells. They are fragments released from large bone marrow cells packed with tools for rapid response. When they encounter a damaged vessel, they stick, change shape, and release chemical signals that recruit more platelets. They also release factors that help the clotting cascade build a stable mesh. Inside platelets are granules filled with molecules that influence inflammation and healing, including signals that encourage nearby cells to begin tissue repair. Because platelets are small and numerous, they can arrive quickly at tiny injuries you never notice. Their lack of a nucleus means they cannot grow or divide, so the body must keep making new ones steadily. Seeing platelets as fragments can sound unimpressive, yet their behavior is decisive. They are first responders, carrying a compact toolkit that can stop bleeding and start rebuilding within moments. Some cells taste glucose levels, then adjust metabolism minute by minute. Your body treats blood sugar like a variable that must be held in range. Specialized cells in the pancreas sense glucose and respond by releasing insulin when levels rise. Other cells counterbalance this when levels fall, helping prevent energy shortages. Glucose sensing is not just measurement. It is chemistry that links fuel availability to signaling. As glucose enters these cells, internal pathways shift, and that shift triggers the release of hormones into the bloodstream. Muscles and fat then respond by taking up glucose, and the liver adjusts how it stores or releases sugar. This coordination can happen repeatedly through a day, tracking meals, activity, and sleep. The fascinating part is that this taste is molecular. Cells do not need a tongue to detect sweetness. They use transporters, enzymes, and ion changes to turn glucose into information. Metabolism is not a background process. It is a responsive control system built from sensing cells. Cells can become stressed, then unfold proteins and trigger rescue systems. Cells live with constant pressure, from heat shifts to toxins to infection to heavy production demands. One common stress is protein misfolding, where newly made proteins fail to reach the right shape. Misfolded proteins can clog the cell's workspace and interfere with essential reactions. Cells respond with rescue programs that slow certain kinds of production, increase helper proteins that assist folding, and enhance cleanup systems that remove damaged parts. If stress becomes severe, cells can signal for stronger interventions, including shutting down risky activity to protect survival. In secretory cells that produce lots of proteins, like those in glands, these stress responses are especially important because the workload is intense even on a normal day. This is a powerful reminder that cells are not brittle. They have built-in crisis management. When conditions turn harsh, cells can reorganize priorities, protect core functions, and attempt recovery instead of simply failing. Aging involves cell changes, including damage, signaling shifts, and senescence. Aging is not one switch that flips. It is a gradual accumulation of changes inside cells and between cells. DNA damage can build. Mitochondria can become less efficient. Communication signals between tissues can shift, altering how inflammation and repair are balanced. Some cells enter a state where they stop dividing and change what they secrete, and that can influence the behavior of nearby cells. The immune system's patterns can shift too, changing how effectively it clears damaged cells and responds to new threats. Importantly, aging does not happen at the same pace in every tissue. Some tissues can renew quickly and mask damage for longer. Others rely on long-lived cells that must maintain themselves carefully over decades. Thinking of aging as cellular change makes it feel more concrete. It is biology over time, shaped by repair, stress, and the slow editing of cellular networks. It is not simply wear. It is a changing conversation inside the body. Senescent cells stop dividing, yet they can influence nearby tissues. When the cell becomes senescent, it enters a protective halt. It no longer divides, which can prevent damaged cells from expanding into something dangerous. Yet senescence is not silence. These cells often release a mix of signals that can affect surrounding tissue, including factors that attract immune cleanup and molecules that influence inflammation. In small numbers, this can be helpful. It can support wound repair and act as a temporary barrier against uncontrolled growth. In larger numbers, the signals can become disruptive, encouraging chronic inflammation and altering how nearby cells behave. The body can clear senescent cells, but clearance can become less efficient with age. Researchers study senescence closely because it sits at the intersection of cancer prevention, tissue repair, and aging. It is a cellular compromise. Stop dividing to reduce risk, but remain biologically active in ways that can reshape the local environment. Cells can be reprogrammed, turning adult cells back towards stem-like states. In the early 2000s, scientists showed something that still feels like a plot twist in biology. With the right set of genetic instructions, a fully specialized adult cell can be pushed back into a flexible state, closer to what we see in early development. These reprogrammed cells can then be guided into different cell types, which offers a way to study disease in a dish using a patient's own biology. A skin sample can become heart-like cells for testing drug effects. It can become nerve cells for studying disorders that are hard to access directly. The deeper wonder is what it reveals about identity. Specialization is powerful, but it is not always permanent. The cell's history can be rewritten with careful nudges, and the new state can be stable enough to grow and differentiate again. This discovery changed regenerative medicine from a dream into an active frontier. Organoids let cells self-organize into mini-organs in labs. If you give certain cells the right support and conditions, they do something astonishing. They begin to arrange themselves into three-dimensional structures that resemble tiny organs. These organoids can develop layered organization, distinct cell types, and even rudimentary functional behaviors, depending on the tissue. They are not full organs, and they are not conscious. Yet they can model key features of the gut, the brain, the lung, and more. Researchers use them to watch development unfold, to study infections in a controlled setting, and to test treatments in tissue that behaves more realistically. Flat cells on plastic. Some organoids are grown from patient cells, which helps reveal why a therapy works for one person and fails for another. The most captivating part is the self-organization. Cells carry enough internal instruction and communication skill to build structure without a blueprint laid out by human hands. In a dish, life rehearses its own architecture. CRISPR lets scientists edit DNA, borrowing tools from bacteria. Bacteria have spent billions of years defending themselves against viruses, and one of their defenses became a breakthrough tool. CRISPR systems can recognize specific genetic sequences and help cut them, which bacteria use to target viral DNA. Scientists learned to guide this system with a designed RNA sequence, aiming the cutting tool at a chosen spot in a genome. Once a cut is made, the cell's own repair machinery can be used to disable a gene or to introduce a corrected sequence. That opens doors for research that used to take years. It also raises serious questions about safety, consent, and long-term effects when edits are attempted in living organisms. In the lab, CRISPR helps reveal what genes do by changing them and watching the consequences. In medicine, it holds promise for certain genetic diseases, and it is already being explored in multiple clinical directions. A bacterial defense became a precision instrument for biology. Single-cell sequencing can reveal cell types once invisible to science. For a long time, tissues were studied in bulk. You would grind them up and measure an average signal, which hides rare cell types and blurs important differences. Single-cell sequencing changed that by letting scientists read gene activity from individual cells, one by one. Suddenly, a tissue that looked uniform became a diverse neighborhood. New subtypes appeared, including rare immune cells, transitional states, and stressed populations that only exist in certain conditions. In the brain, this approach has helped map incredible diversity across neurons and support cells. In tumors, it can reveal which cells are resistant, which are vulnerable, and which are interacting with the immune system. It also helps track development because you can see cells moving through stages instead of only seeing the endpoints. The wonder here is granularity. Life is not always made of a few neat categories. Many tissues are mosaics, and understanding health often depends on noticing the quiet minorities that bulk methods miss. Cells communicate through tiny vesicles, sending packaged messages outward. Cells do not only communicate with dissolved molecules that drift away, they also send physical packages. Tiny membrane bubbles can bud off from a cell, carrying selected cargo inside. That cargo can include proteins, lipids, and pieces of genetic material wrapped safely for travel. When another cell takes up the vesicle, the contents can influence behavior, changing gene activity or signaling pathways. This is a different style of communication. It is not a simple shout into the bloodstream. It is more like shipping a sealed envelope with specific contents. Vesicle messaging appears in immunity, in tissue repair, and in cancer biology, where tumors may use vesicles to influence their surroundings. Researchers are also exploring vesicles as delivery vehicles for therapies, because nature already knows how to move molecules between cells without tau, destroying them. The fascinating idea is that cells can export curated parcels of information, and those parcels can reshape distant targets in a way that feels almost like biological mail. Exosomes can carry RNA and proteins, influencing distant cells. Exosomes are a particular class of very small vesicles that can travel through body fluids and act as long-range messengers. They can contain RNAs that affect which proteins a recipient cell makes, and proteins that can trigger signaling changes on arrival. Because they are wrapped in membrane, their cargo is protected from being broken down too quickly. That makes them useful for communication across distance, not only within a single tissue. Scientists study exosomes as possible biomarkers because their contents can reflect what a hidden tissue is doing. A tumor, for example, may release exosomes with a distinctive molecular signature that could be detected. Exosomes also complicate biology in a thrilling way. A cell's influence is not limited to hormones and nerve signals. Cells can release traveling packages that carry complex instructions, and those instructions can change behavior in places the original cell will never reach. It is molecular influence with a passport. Some cells survive low oxygen by switching to emergency metabolism. Oxygen is central to efficient energy production, but tissues do not always get a steady supply. During intense exertion, in poorly supplied regions, or in certain diseases, oxygen can drop. Cells respond by shifting how they generate energy, leaning more on pathways that do not require oxygen, even though those pathways produce less energy per liter molecule of fuel. This emergency mode helps cells stay alive through temporary shortages. It also changes the chemistry around them, because byproducts can accumulate and alter the local environment. Some cells slow down non-essential tasks to conserve resources. Others increase transporters to pull in more glucose, trying to compensate for reduced efficiency. This switch is not a sign of weakness. It is a survival adaptation built into the operating system of cells. It also has medical significance, because regions with low oxygen can behave differently during injury and disease. When oxygen fades, cells do not simply fail. They improvise. Cells can cooperate, but they also compete inside tissues. A healthy tissue depends on cooperation. Cells share signals, respect boundaries, and specialize for the common good. Yet tissues are also environments where resources are limited and cells can compete for space, nutrients, and survival cues. Most of the time, the body's rules keep competition fair. Damaged cells are removed. Overcrowding triggers stop signals. Immune patrol catches many cheaters early. When these controls weaken, competition can turn harmful. A cell that ignores the rules can outgrow its neighbors and reshape the tissue around it. Even outside cancer, competition can appear in subtle ways, like when certain cell populations expand after injury and crowd out others. Biology is both community and selection. That tension is part of what makes tissues adaptable. It allows the best-suited cells to repopulate after damage. It also creates risk when regulation fails. Understanding this balance helps explain why health is not only about individual cells being strong. It is about the rules that govern how cells share a living space. Life's unity shows in cells because all share the same basic toolkit. A human cell and a yeast cell are separated by vast time, yet they still rely on shared fundamentals. They use DNA to store instructions. They use RNA and ribosomes to build proteins. They use membranes to separate inside from outside, and they use enzymes to speed up chemistry. This unity is one of the most comforting ideas in science. It means that studying a simple organism can reveal truths that apply to complex bodies because the core machinery is conserved. It is why bacteria taught us about gene regulation, why yeast taught us about cell division, and why fruit flies helped reveal developmental principles. Evolution changes details, adds layers, and invents new structures. Yet it rarely throws away the essentials that work. When you learn cell biology, you are seeing the shared operating system of life on Earth. The same basic parts are being reused, refined, and recombined into every creature you know, from microbes in soil to mammals in cities. Different forms, shared foundations. Every breath supports cells, and every cell quietly supports you. Breathing can feel like a single action, but it is really a chain of cellular events. Oxygen enters the lungs, crosses thin barriers, binds to carriers in blood, and is delivered to tissues that need steady energy. Inside cells, that oxygen is used to extract more energy from fuel, supporting everything from muscle contraction to brain activity. At the same time, cells produce carbon dioxide as a waste product, and that gas must be carried back to the lungs to be exhaled. This exchange is constant, and it is coordinated across organs and cell types that never meet, yet depend on one another. If one link weakens, the whole system feels it. What makes this so awe-inspiring is the scale of cooperation. A breath is not only air moving in and out. It is life-sustaining chemistry being fed and cleared, moment by moment, across trillions of living units. Your body is a partnership between the atmosphere and cellular metabolism, renewed with every inhale. As we come to the end of our journey through cell biology, you can let the ideas settle like dust motes in a beam of morning light. Tonight, we stepped into the smallest living rooms of life. We wandered through membranes that choose what may enter and what must stay out. We visited nuclei that keep the long memory of DNA, and we listened to cells as they sent signals, built proteins, moved with purpose, and repaired what time and stress can fray. We watched cells divide with care, and we noticed the quiet courage of systems that protect the whole, from immune patrols to the careful limits that keep growth in check. We even glimpsed how life learned to become complex by partnering, specializing, and building tissues that hold together like woven fabric. And now, you do not need to hold on to any of it. If your mind is still turning, let it turn slowly. Like a microscope knob being eased into focus, then eased back again. Let your breathing find its own comfortable pace. Feel the weight of your body resting where it is. Allow your jaw to soften. Allow your shoulders to drop. There is nothing to solve, nothing to remember perfectly. If you enjoyed this exploration, you're welcome to like, subscribe, or share a thought below. It helps others find their way here too, one sleepy soul at a time. And if you happen to still be awake, there will be another video waiting for you on screen, ready to carry you onward through another quiet corner of science. But for now, there's nothing else you need to do. Let the images fade. Let your breathing soften. Allow the sense of depth and quiet time to settle into stillness. The pressure eases. The light dims. And the world can wait until morning. Sleep well, and good night.